So to get an overview of these methods which I just mentioned, especially on array CGH and SNP array, here is a comparison of a so-called Cree-Duchat syndrome. I hope many of you might already have heard of it. It's quite frequent monosomy of a very telomeric part of the chromosome 5 on this. As you can see here, you can easily detect this this aberration with classical karyogramming. So cytogenetics, you see here the missing part. So this for this very large deletion, this method is still usable. The second method which could be used for this purpose, for example, would be FISH analysis. So FISH stands for fluorescence in situ hybridization. And here you have fluorescently labeled probes from this specific region, and you just hybridize it onto the chromosome in the metaphase as it's shown here, or also in the euchromatic karyotype. And you should see here, if you would normally see 4 signals, 2 green and 2 red in a normal karyogram, but here you see 1 is missing because the probe from the very telomeric here is not present in the deleted chromosome. So that's why you see only 3 signals here. Another method which is not in use anymore here in our lab is CGH, which stands for comparative genomic hybridization, which would also be a very highly resolution method for detecting this abnormality here. As you can see here, this split signal between red and green is shifting to red because of the deletion, and that's why you can say, okay, here we have a deletion located on this chromosome. And the current method, which is actually already on the decline in our institution because it's shifting to another method, which I will present later, was array CGH, where you have probes all over the chromosome. You see each dot is representing 1 probe, and you see in the region of this Cree-Duchat on chromosome 5, you see a deletion here, and the probes are shifting to the left side here, which represents a deletion. So you see clearly many methods would be able to detect this rather large deletion, but when it comes to smaller deletions for sure, or insertions, whatever, then the karyogram will not be suitable anymore. The FISH method would only be very specific. If you look for a certain deletion or insertion, it's very useful, but you miss out any other things which can be present. CGH is, as I said, not in work anymore in most of the labs, also in ours. And array CGH is a very highly resolution method to detect imbalances on the genome wide. But we think 5 years ago we switched to around this years we switched to Infinium CytoSNP 850K assays, which is a SNP array, which also gives us the opportunity, which is a high resolution method to detect imbalances on the genome. You see the highest resolution would actually be 1 kb, which is really very good in regions with clinical relevance. In this case, here you see the probes which are located in an exome of a gene, and so many more probes are located in this method in the relevant, clinically relevant regions, and not so many are located in regions without any clinical relevance. So it's not a linear distribution of all the SNPs, but you have more SNPs in regions where where findings can be very crucial and is important for health, and where you don't have any genetic information which is translated. For example, you have a very small number of SNPs. Used means you have covered every region which is translated quite frequently, and you can actually see abnormalities of copy numbers in this data set as well. So you don't have to have whole exome sequencing and array CGH. You can really do both analysis, copy number, and SNP variations within whole exome sequencing technology. So you can see here, this would be a normal distribution of reads, as they say, as we call them here in the next generation sequencing, whole exome sequencing. And in a patient who has a deletion in this region, for example, you see almost half of the reads in the region, which means that here is the genomic region is deleted on one of the two alleles. So the same as another example of for this on this in this gene, you see the normal read depth in this patient, but in the patient here you see a diminished number of reads in this region, which means that this exon and the following intronic region seems to be deleted. So the same is, of course, you can also see insertions as well. It's not so good as deletions, of course, but still we are able to detect it and do some diagnostics out of it. We now have come to a status where we report deletions if the deletion is spanning two consecutive exons. We are able to find it precisely. And additionally, we look closer to exons. If we see deletions in only one exon in a gene, which is very likely for a special phenotype, then we also validate this deletion or a presumable deletion with real-time PCR to verify or falsify the initial finding. So that's how we proceed normally today. What is not possible with this sequence is, of course, if you have any reciprocal translocation, for example, it's still not possible to detect this, and we have to really rely on karyotyping to really find reciprocal translocations at the moment. But as a future perspective, I can also say that we probably in 1 or 2 years will switch to whole genome sequencing instead of whole exome sequencing, and as you see on the right side, then we would be able to also detect reciprocal translocations where there's no genetic material missing. But you see, because you have split reads here, you can easily detect the right breakpoints where two chromosomal parts are fused together. So this is also a very nice perspective, and leads to a higher detection rate of such translocations. So this is now a picture of our molecular lab. As you can see here, we have tons of PCR machines who is doing the PCR work, which is needed for whole exome sequencing approaches. And here are our next generation sequencing platforms and the Sanger sequencing platform we use for mutation detection. So on the left side, you see the Life Technologies machines, which you all might be very familiar. So this is a 16 capillary sequencing machine, and this is a 48, I think, a capillary system. And these are the next generation, one of our next generation sequencing machines, which allows us to perform mainly whole exome sequencing. So just to give you a small introduction on Sanger sequencing, also I'm pretty sure you might be familiar with the technique. So this is based on the invention of Frederick Sanger, who is who died in 2013. He has actually got 2 Nobel Prizes in science. So he's was quite a bright guy, to be honest. And he was able to introduce us to this nice method. There is still the golden standard for mutation detection, still actively used in our institution. And you see here a capillary system, as it's when you open the machine, and you see you can parallelly sequence 16 PCR products normally, and you get, in 1 hour, approximately, I would say, 16,000 KB of sequence information. So this is all incorporated in this machine. And if you compare this now to next generation sequencing, just to give you a I want to illustrate it, how much more information, cDNA information or genetic information you get out of one run is illustrated here. So each of these dots represents a cluster, which is then later on leading to a sequence file. We call it a read. So it can be compared, one of these dots can be compared to one of these capillaries roughly. And the sequencing itself takes place in such a flow cell, as you all, or many of you might have already seen. We use Illumina technology, which is the main supplier for these technologies. And how does this work now? So for each cycle, the nucleotides which are labeled are put into the flow cell, and they bind wherever it finds a corresponding template strand, and then a photo is done for the whole flow cell and recorded, and this gives you the sequence information, which is then leading to hundreds of millions of reads, which finally then annotated to the human reference genome. So here you see the same dots, the same after 5 cycles, I think. So you see here the different bases which are incorporated here on during the sequencing process. So these are roughly, let's guess, 30 sequencing reactions in parallel, what you see here. But this only represents, so here you can, these are the bases which can be seen for this one cluster, which is included in the read information later on. But this small field is just this part of the flow cell, and this later on is just one small part of this flow cell consisting of 8 rows. So you can roughly estimate how much information you can obtain from one next generation sequencing run. So this gives you much more opportunities in the diagnostic and scientific approach as well, of course, which we, of course, make use in our institution as well. So if we just compare it, we have on the left side the classical Sanger approach leads to 16 parallel sequencing reactions per hour versus 150 million parallel sequencing reactions per run on the next generation sequencing machine. But this is only, it gets more and more because the machines are getting bigger and bigger, and each generation offers more and more capacity. So, our latest machine here is the Nova 6000, which enables us to do whole genome sequencing on a daily basis, actually, to a quite cheap price. So we are capable in the future of doing whole genome sequencing in diagnostic approaches as well. But for the moment, we are still doing whole exome sequencing for most of the questions we have to answer from the patient's perspective. Only a little bit we use these smaller machines for targeted panel analysis, mainly in the cancer genetics, because here we know all the major genes which are responsible for hereditary cancer syndromes, and this is a very a cheap approach to gather this important diagnosis for the patients. So just to give you a brief overview of what is our portfolio of diagnostics, of genetic diagnostics in our institution. We cover eye disease, connective tissue diseases, intestinal diseases, morbid diabetes. We have panel also for neurogenetics. So these are all so-called soft panels. So we analyze the whole exome, but the further analysis of the variants are restricted to the variants which are in association with the phenotype which is present. A huge part of our diagnosis is also focused on tumor genetics, as you can see here, mainly BRCA1 and 2, so breast cancer gene 1 and 2 diagnostics, which we cover by panel analytics, as I told you, and but also, which is really on a rise right now, we analyze DNA of circulating tumors, which enables us to monitor tumors, which is very relevant for the applied therapy for certain tumors. So this is very important future direction which we have began to I think 10 years ago, but we are really, I would say, very special last year in Graz on this special method. And that's why these are in this suspicion of genetic syndrome is still in not in red, because it's not based usually on next generation sequencing. So, but the analysis is still done classically with cytogenetics and sometimes with SNP array, but more and more, of course, as well on whole exome sequencing. And here, especially when it comes to genetic syndromes, in the prenatal situation, we are doing trios. So we are sequencing the parents and the fetus, which enables us especially to detect de novo mutations, which are quite frequent when it comes to genetic syndromes. And of course, moreover, we have very specific familial situations, specific questions, which where, for example, familial mutations are known and has to be done at the additional family member, which are usually covered by Sanger sequencing. That's why I didn't color this specific question here as well, not in red. So this is a brief overview of what we are doing or can offer to the patients, and how the methods are implemented in what we are doing. And as my last slide, actually, I want to show you a platform for validating specific variants, which you might, many of you might know or not. That's why I just wanted to express my thought about Franklin, the Franklin platform by Genox, which I think is a very excellent tool to validate certain candidate variants of whole exome sequencing approaches. For example, Musamil might know this variant because he has prepared one publication with his students, and you get all the information about which is available in certain on all the databases which are relevant in one page actually, and you can click on everything and see whether this is a suitable candidate for your phenotype or not. So I can highly recommend you to use it. It's free, open for everybody. So you can upload your VCF file from the whole exome sequencing and analyze your results in a really, really nice way. And yeah, so that's everything from my side for now. And I hope it was not too confusing for you, but this is quite difficult to get to deliver an overview of my institution in such a short period. But feel free for asking me some questions. I hope you enjoyed the lecture so far. Thank you. Hello? Hello?
Yes, Christian. Thank you very much for your very nice presentation. It was really very informative. And yes, can you hear me?
Hello?
Yeah, can you hear me? I can hear you. Can you hear me?
Hello?
Yes, Christian. Can you hear me now?
My audio is not working. Not sure if you can hear me, or
Yeah,
I hope you could hear me.
I can hear you, and my microphone is also on.
Hello?
Yeah. Can you hear me now?
Hello? Hello?
Yeah, I can hear you.
Yeah, I can hear you now.
Okay, perfect. So thank you very much for your very nice presentation. And now I would like to open the session for the question. So now I would like to invite the audience if they have any question. And I also have few very quick questions, but I will ask later. First, I will allow the audience to ask the question. So anyone from the audience, if they have any question from the Christian, please go ahead.
Hello, Dr. Christian.
Yes.
Hello?
Yes,
can you speak up a little bit?
Yeah, can you hear me?
I can hear you.
Okay. Thank you for the nice presentation. It was really informative. I have one question. Do you perform expression analysis in your lab?
Yes, we do occasionally, especially when it comes to splice site mutation analysis. Then we do it. So we either we do mRNA analysis, or if it's not available, we also do sometimes, but it's more on a scientific approach, mini gene assays, things like that, to verify any effect on splicing on the expression. So that's what we normally do in our routine work.
Okay, thank you.
You're welcome.
Yes, anyone else who has any question, please go ahead. We have limited time of the session. We have just left with 7 minutes, and then the session will close automatically. So I would request the audience to quickly ask the question, if they have any. Or maybe I will ask a very quick question. Christian, in terms of next generation sequencing, we have whole exome sequencing and next generation whole genome sequencing. So
yes.
In terms of diagnosis, which one is more appropriate, the whole exome or whole genome?
At the moment, we are only offering a whole exome sequencing, because it's not the whole genome sequencing is not yet covered by the health system.
Okay.
So we do not offer it right now. We could offer it theoretically, because it's with our machines, but we do it. I guess the switch will be, let's say, in 2 years probably. Then I would estimate then we will switch from whole exome sequencing to whole genome sequencing, because you have a high yield of positive results. Let's say that you don't miss out of any interesting intronic variant, or also you can detect CNVs very, very well with this technique. So the future is for sure whole genome sequencing.
Okay.
Whole exome sequencing is only a transition time.
Okay. And my next question is that if you talk about the different techniques, if we even talk on the cytogenetic techniques, the molecular cytogenetic techniques, and then if you talk on the molecular genetic techniques, which technique is recommended? I mean, in terms of the genetic counseling, that which technique is recommended?
Sorry, I didn't get the last sentence.
Yeah, so
I didn't get the last sentence.
Yeah, I was talking that we have different molecular genetic techniques, like the cytogenetics, and the molecular genetics, like the sequencing techniques. So based on which parameters we recommend, the genetic counselor recommends different techniques. Either they recommend the cytogenetics or they recommend the molecular genetics. So what are the parameters who is going to decide that which technique is more appropriate for the genetic diagnosis?
It depends largely on the situation, which phenotype is present, if, for example, it's absolutely precisely phenotyped by a clinician that it's, for example, cystic fibrosis, then you probably will not make whole exome sequencing. We will do a kit for the mutation kit for CFTR. You know, it's depending on what is known on the clinical side. If it's very heterogeneous genetic disease, for example, a peripheral neuropathy, for example, then we will do for sure whole exome sequencing. But just my guess is from that 90% of what we are doing now is whole exome sequencing. Okay. And everything else is maybe 10% in the molecular genetic department.
Okay. So anyone else from the audience?
And we just then, it's, yeah.
Yeah, carry on, please.
So it's, if we do whole exome sequencing, we are not always analyzing the whole exome, but we are restricted to this, as I told you, to the soft panels, where we have a gene list which is responsible for a certain disease, and only these are really analyzed. And we guarantee here that it's 100% covered and nothing is really missed in the sorry, no exon is missing really in the diagnosis. For remaining genes, for example, we only offer it as a screening purposes, where we really don't have a look at every single exon, if it's covered or not. We just screen them.
Okay.
So that's the difference.
Okay.
And also, what I maybe we should also add. We also use HBO terms for genome-wide search of any mutations. If it's not really clear which syndrome it is or which disease, then we use HBO terms, which helps us prioritizing the variants which could be responsible.
Okay, perfect. Thank you very much. So anyone from the audience, if they have any question, please go ahead. Otherwise, I'm going to close the session because we have only left with 2 minutes.
Hello.
Yeah.
Yes.
Can you, sir, your presentation was so good. My question is that can we remove genes from DNA at any stage of life for the protection of cancer from future?
Actually, I didn't get it.
Sorry.
Please. I heard that.
Mustamil, did you hear? Can you tell me what?
Please repeat your question so that I may elaborate.
Yes, sir. I said that can we separate cancerous genes from the DNA at any life of stage, at any stage of life, so it cannot cause cancer in future?
I'm not sure if I got it. If we separate cells from cancer, you mean?
No. Probably
yes,
he's actually want to ask that if can we rectify the DNA, or can we remove the defective DNA from the cancerous cell in order to? Okay.
No, that's not the purpose of what we're doing. We only see if the tumor is evolving in the course of this on the molecular level, and these aberrations and mutations of tumor we can detect, and from that result we can different thoughts of therapy which might be promising. So this is called personalized medicine, which is very cost and labor intensive, but that's where the way goes in cancer diagnostics.
Okay, Christian, thank you very much. So that's it for today's session. I'm really thankful to you for sparing the time and for the very important topic.
It was my pleasure, really. It was
thank you very
great
much.
to having me. And,
so
yeah,
the session may close in a minute because we have already completed our duration. So thank you very much, and I'm hoping for the next session, maybe on some other times. So thank you very much.
Thank you very much all to all the students, and I continue. Hopefully, have a continually nice vacation. So
Yeah, hopefully. Thank you very much. Have a nice time.
have a good day, everybody. Goodbye.
Thank you very much. Goodbye.
Goodbye.